Hi folks, this is Dr. B. Sivaramakrishnan from Department of Genetic Engineering, SRM Institute of Science and Technology. I welcome you all for this presentation on human chromosomes, structure and organization. What exactly is a chromosome and where is it basically present in a cell? Chromosomes are thread-like chromatin structures and they are present particularly in the nucleus of a cell. Chromosomes, and particularly chromatin structure, invariably is said to contain both the DNA component and histone component. The histone component basically represents the protein one, and there are also certain kind of non-histone component proteins also, which are an integral part of the chromosomes. They were first observed as thread-like structures by a German biologist named as Walter Fleming in the year 1882. The term chromosome, however, was first coined by Waldeyer in the year 1888. They are said to contain the genetic information which is carried on by the DNA, and it is vital for the cell function and as well as inheritance. If you look at the structure of a chromosome, typically it is said to contain a shorter or a smaller arm, and it is also said to contain a longer arm. The shorter arm is called as the P arm, and it is otherwise also known as the petite arm. The longer arm represents the Q arm, and it is also abbreviation is can be expanded as actually the QUEUE. The Q arm, it is also called as the longer arm. Chromosome, since is if you are actually looking at it, it's the chroma component is actually represents the colored component, and soma represents the body of the chromosome. And due to their marked affinity for the basic dyes like GIMSA, we actually use these dyes for actually understanding the banding pattern present in the chromosomes. Human chromosomes are fascinating. In every cell, it is said to contain 46 chromosomes, and these 46 chromosomes are organized as 23 pairs. Broadly, to represent these 46 chromosomes can be divided into two categories, either as the autosomes or the allosomes. Autosomes represents the non-sex chromosomes. As you can see in the ideogram shown over here, you can actually observe the G banding pattern being represented for all the 23 pairs of chromosomes. But the autosomes represents the chromosomes from number 1 until 22, whereas the allosomes are the sex chromosomes, and in the case of males it is XY chromosome, or it is actually in the case of females represented as the homogametic sex with the 2X chromosomes. If you check out for the offsprings that are arising from the fertilization of a male and a female gametes, you can actually observe that it could be either a 50% probability of or a chance of occurrence of a male, or 50% chance for occurrence of a female. There are three features that are available to study and as well as classify the different types of chromosomes. The three key features that enables us to identify these similarities or the differences between the chromosomes is based on their size or the banding pattern, as I spoke earlier with regard to the staining because of the basic dyes like GIMSA, or the third component could be the centromere position of the chromosomes. Centromeres are nothing but the regions in the chromosomes which are said to be densely packed, and they are said to be appearing only in the center position predominantly, though they can also vary in their position where they are actually placed. Chromosomes can differ in the positioning of the centromere, the place on the chromosome where the spindle fibers are attached during the process of cell division. In general, the centromere is present near the middle, then the chromosome is considered as metacentric. If the centromere is present towards closer to one end, then the chromosome is considered as acrocentric or submetacentric. If the centromere is present at the poles or at the near end of the chromosome, then we consider it as telocentric. Here we have tabulated the 7 groups of the chromosomes based on their size. The chromosome number 1 until the 22nd chromosome invariably has been categorized in such a manner that they are the autosomes which are numbered from the largest to the smallest, except that chromosome number 21 is little bit smaller than the chromosome number 22, though it is placed little bit ahead. And Y happens to be the smallest chromosome, which is also said to contain numerous microsatellites present in them. We were able to differentiate the chromosomes based on their size and as well as the position of the centromere. In the chromatin, thread-like structures which are present in the chromosomes can be further categorized as heterochromatin or euchromatin. What are heterochromatin? Heterochromatin represents the darkly stained regions which are said to be AT rich. Euchromatin represents the lighter stained regions which are said to be GC rich. And this kind of staining procedure is basically attained because of usage of the basic dyes which interact with the phosphate negatively charged groups of the DNA molecule. The darker bands can also be observed near the centromeres as well as the telomeric ends of the chromosomes. The less dense and as well as the more condensed regions represent the euchromatin and as well as the heterochromatin respectively. You can observe here in the figure A component which represents the TEM image, wherein you can see the presence of euchromatin, which are the less dense regions, and the darker condensed regions represents the heterochromatin and as well as the nuclear heterochromatin regions. In figure B, you can observe that these set of euchromatin, some part of it being basically condensed and some regions basically present as the loosely packed or the little bit less densely packed. And this you can basically observe only in the case of the euchromatins, whereas in the case of heterochromatins, they are basically said to be highly condensed. Why is this basically occurring? Do you know the length of DNA present in the cell nucleus? It is something like around 2 meters of the DNA is totally wound and then coiled and as well as densely packed within a small region of just something like around 6 micrometer of the cell nucleus. A cell, an average cell, in the case of the humans, you can basically consider it anywhere between 10 to 30 micrometer. And if you consider the size of the or the diameter of the nucleus alone, it is just 6 micrometers. You can imagine or how fascinating it is that such a huge length of DNA up to 2 meters is coiled and densely packed within the nucleus of just 6 micrometers. For this to be achieved, the human's chromosomes have an organization phenomenon. And this actually happens at 4 levels. The first level, it involves the nucleosome formation. Second level involves the solenoid formation. The third level involves the looping formation, and as well as the fourth level, at which due to the one of the mitosis metaphase chromosome phenomena, which leads to the complete condensation of the chromosomes. You have now seen the difference between the euchromatin and as well as heterochromatin. Why basically certain part of the gene segments or the chromosomes are said to be highly condensed, and why do you need to be little bit relaxed of these chromosomes to be relaxed in certain case scenarios? The reason can basically be could be due to the possibility gene regulation that could be achieved by such kind of activity of the heterochromatin and as well as the euchromatin conditions. When the DNA is said to be less densely packed, this will favor the binding of the RNA polymerase to the DNA segments of the chromosome, resulting in the transcription occurrence, one of the molecular biology phenomena, which leads to the expression of the segments of these genes. But whereas if the DNA is not required or the gene expression is basically not required, then in that particular case scenarios, they could be condensed and the exposure of the DNA to the RNA polymerase can be avoided. The first level of structural organization of chromosomes are called as nucleosomes. What are nucleosomes? These are 10 to 11 nanometer chromatin fibers. They are said to be made up of both DNA and as well as histones, as we basically see over here. You find that certain length of DNA is basically wound around the histone octamer. And when I say the histone proteins, which are said to be generally positively charged, and you know that the DNA, due to the presence of the phosphate backbone, is said to be negatively charged. This results in actually an interaction between the DNA and as well as the histones. There are 5 different types of histone proteins. They have the subtypes include H2A, H2B, H3, and H4, which is the more predominant ones. And these are the duplicated ones, or I would say the tetramer moieties. And when two tetramers of these histone subtypes join together, they actually form an octamer complex of your histones. Whereas there is another subtype, or the fifth subtype, which we call it as the linker DNA. Now, if you consider the size of the DNA, invariably it is just only 2 nanometer. A DNA, when it has to be bound, it goes 1.65 times, turning around the octamer histone moieties. This DNA that is bound around the octamer of histones is called as the core DNA, whereas the linker DNA is the DNA which connects two octamers of nucleosomes along with the DNA. Okay, the region in between these two nucleosomes where the DNA is connecting it, that actually represents what is called as the linker DNA. As you can actually observe in this particular image over here, you can see the octamer of 4 histones, which are twice in number, and invariably that is the DNA that is bound 1.65 times around the histone octamer is held together basically by the H1 subtype of the histone. And you can also see the same linker DNA being represented between the two components of your nucleosomes, wherein which is both the nucleosomes are separately held together by the DNA from falling apart by the respective linker H1 components of histones. This represents the first level of packaging of the DNA, or the folding of the DNA. When nucleosomes are visualized under the microsome, we basically observe it as a string of beads, wherein the beads represents the histones and the string represents basically the DNA that is wound around it. Here we have also tried to show you the three-dimensional and as well as the two-dimensional image of the uh, nucleosome units. You can very well appreciate that the DNA, which is said to be double helical by nature, is just 2 nanometer, which goes 1.65 times around the histone octamer entities, and which is also held together by the H1 component of the histones. The linker DNA is just only around 50 to 90 base pairs in length, whereas the core DNA, which is bound around the histones octamer, is said to be around 146 base pairs in length. The role of the H1 histones is not only to protect the DNA that is bound around the octameric uh, histones in a tight manner, but also they can interact with additional H1 aggregates, thereby establishing a 30 nanometer solenoid fiber structure, resulting in the second level of packing of DNA. In earlier two case scenarios, the folding of the DNA was basically achieved due to the activity of the histone proteins, whereas in the third level of packing, this is achieved due to the activity of non-histone proteins, which serve to provide the scaffold over which the chromatin fibers can actually fold and as well as coil, in establishing a 300 nanometers of chromatin fibers, which represents the higher order structure of packaging of the chromosomes. The fourth level packaging of the chromatin happens due to the metaphase chromosomes. Loop domains they further coil and fold, and this results in compacting the chromatin, resulting in the characteristic metaphase chromosomes as observed in the case of karyotyping procedures. Such packing occurs during the mitosis phase of the cell cycle. It must be fascinating for you to understand how 2 meter long length of DNA is basically compacted and packed within a small region of 6 micrometer of the nucleus. As you can basically observe, the DNA double helix, which is actually the 2 nanometer in size, is folded and wound around the histone octamer units and held together by the linker H1 histone protein components. This is further then made as a hexamer or 6 units of these nucleosomes are joined together further by the histone proteins, particularly the H1, which aggregates and thereby resulting in further compacting of the DNA. At the third or the tertiary level, you can observe furthermore scaffolding and as well as coiling of the DNA is basically, or the chromatin is basically observed. This results in generating the 300 nanometer fibers. Further, this is furthermore compacted and then results in generating the 700 or to up to around 1400 metaphase chromosomes condensation. Overall, these levels of compaction aims to provide a kind of a balance and as well as a functional accessibility. We have seen the role of heterochromatin and as well as euchromatin and their importance in the regulation of gene expression. DNA packaging enables the cell to fit such vast amount of genetic material within the nucleus, thereby protecting it from damage and as well as regulating the expression of genes efficiently. I thank everyone for patiently listening.